Inside the arterial wall, lipid deposits slowly accumulate, thickening over years into an obstructive atherosclerotic plaque. We have treated this mechanism for decades with daily statins and monthly PCSK9 inhibitors. Yet many high-risk patients maintain dangerously elevated levels of atherogenic lipoproteins, despite maximal medical therapy. Suppressing low-density lipoprotein, or LDL, only solves part of the equation. Triglyceride-rich lipoproteins circulate concurrently, continuously driving atherogenesis and leaving patients exposed to significant residual risk. The mechanical limit of our current lipid-lowering pharmacotherapy lies in its delivery. Efficacy requires lifelong, unbroken adherence to a regimen of daily pills or routine injections. This chart plots patient adherence over a 12-month period. We see a steep decay across oral and injectable therapies as months progress. Missed doses cause circulating drug concentrations to plummet. Once curves drop below this minimum effective threshold, therapeutic protection collapses. Bypassing human behavior requires a different class of intervention. We are looking at the exact biological mechanisms required to treat cardiovascular disease with a single, permanent dose. The blueprint for this intervention exists in the translation of a rare genetic discovery regarding the ANGPTL3 protein into CTX310, an in vivo CRISPR-Cas9 therapy. Understanding this requires isolating the target, mapping the engineered delivery vector, and analyzing phase 1 clinical data. Resolving refractory metabolic disease requires rewriting hepatocyte DNA to shut down lipid accumulation at the source. The liver synthesizes and secretes a protein called ANGPTL3 into the bloodstream. Physiologically, it functions as a master brake on systemic lipid clearance. This diagram outlines ANGPTL3's structural anatomy. Critical regulatory components are at the N-terminus, flanked by the furin cleavage site and the SE1 domain. The SE1 segment acts as the primary docking unit required to bind with lipoprotein lipase, or LPL. Inside the capillary lumen, ANGTNPTL3 complexes with a sister protein, ANGTNPTL8. Together, they physically bind to circulating LPL. This binding triggers a severe conformational shift. ANGTNPTL3 forces the hydrolase domain of LPL to unfold, converting an active enzyme into an inactive monomer. Inactive LPL ceases to hydrolyze triglycerides, creating a physiological bottleneck. Triglyceride-rich particles rapidly accumulate in the blood. We know this break can be safely removed because of human genetic evidence. Epidemiologists identified specific families carrying natural loss-of-function mutations in the ANGTNPTL3 gene. These carriers present with a phenotype known as combined hypolipidemia. Their blood panels show profoundly low levels of both triglycerides and LDL cholesterol. Clinical follow-ups reveal a highly protective safety signal. These genetic knockouts exhibit a drastically lower risk of coronary artery disease, while showing absolutely no adverse physiological side effects from the missing protein. Nature has already mapped the exact mutation required to achieve lifelong lipid clearance safely. The clinical challenge is strictly mechanical, engineering a therapy that replicates this exact genetic knockout. Pharmacological interventions like monoclonal antibodies require endless readministration to suppress circulating proteins. Genome editing cuts off production directly at the source. CRISPR Therapeutics developed CTX310 as an in vivo CRISPR-Cas9 therapy, relying on a lipid nanoparticle, or LNP, delivery platform to target the liver. This diagram reveals the LNP architecture. A synthetic lipid shell encapsulates two critical payloads, folded Cas9 messenger RNA and an ANGPTL3 targeting single guide RNA. Upon reaching the liver, the lipid nanoparticle fuses seamlessly with the hepatocyte's lipid bilayer, depositing the engineered RNA directly into the cytoplasm. The hepatocyte's endogenous ribosomes intercept the delivered messenger RNA. They translate the instructions, synthesizing the Cas9 endonuclease protein from scratch. Once synthesized, the Cas9 protein physically binds with the single guide RNA, assembling into an active ribonucleoprotein complex. Guided by nuclear signals, the complex enters the nucleus. The guide RNA locates the ANGPTL3 locus, and Cas9 cleanly severs both DNA strands. The hepatocyte initiates rapid repair, introducing small insertions or deletions. This creates a permanent loss-of-function mutation. The exact pharmacokinetic profile of this delivery system ensures the Cas9 mRNA and the resulting protein are rapidly degraded by the cell shortly after the edit is complete. Transient expression of the editing machinery eliminates the risk of long-term off-target mutations while leaving behind a permanently silenced ANGPTL3 gene. The first-in-human Phase 1 clinical trial for CTX310 tested this mechanism in an open-label format. The cohort consisted of patients with severe hypertriglyceridemia and homozygous familial hypercholesterolemia. These are individuals completely refractory to the standard of care. This chart captures the primary biomarker response. At the highest therapeutic dose of 0.8 milligrams per kilogram, patients achieved a mean reduction of 73% in circulating ANGPTL3 protein, with maximum individual drops reaching 89%. Erasing the protein from the bloodstream immediately transferred to the patient lipid panels. At that therapeutic dose, triglyceride levels fell by a mean reduction of 55%. Maximum individual reductions recorded drops as deep as 84%. For patients entering the trial with dangerously elevated baseline triglycerides above 150 milligrams per deciliter, the mean reduction held at a strict 60%. The secondary efficacy endpoints tracked LDL cholesterol. That metric showed a mean reduction of 49%. For 2 participants already receiving background PCSK9 inhibitors, the therapy achieved greater than 80% reductions in LDL from their baseline levels. Across all cohorts, the safety profile remained strictly stable. The infusion was well tolerated, producing 0 dose-limiting toxicities and 0 treatment-related serious adverse events. Hepatic safety was rigorously maintained. Despite the lipid nanoparticles specifically targeting the liver, clinicians observed no grade 3 or higher changes in liver transaminases. The data from this Phase 1 trial proves the mechanical viability and safety of deploying in vivo CRISPR editing directly into the human cardiovascular disease pathway. This diagram outlines the systemic landscape of lipid metabolism. It explains precisely how CTX310 forces simultaneous drops in both LDL and triglycerides. By applying genetic intervention at the CRISPR base editing node, the therapy permanently severs the inhibitory signal radiating from ANGPTL3. Without that molecular break, lipoprotein lipase and endothelial lipase are left in a perpetually disinhibited, highly active state across the vascular system. This hyperactive lipolysis strips triglycerides from VLDL and chylomicron remnants, clearing them from the bloodstream entirely before they can be metabolized into dense LDL particles. For patients with homozygous familial hypercholesterolemia, this solves the core biological defect. It clears atherogenic lipids through a pathway that is entirely independent of the LDL receptor. Traditional statins and PCSK9 inhibitors force the liver to upregulate LDL receptors. If a patient genetically lacks functional receptors, those drugs fail. CTX310 bypasses the receptor entirely. This biological bypass extends beyond rare genetic disorders, offering a direct solution for the millions of ASCVD patients battling adherence fatigue and residual risk profiles. The clinical execution of ANGPTL3 targeting proves that lifetime lipid control no longer requires daily medication. A single targeted edit can permanently correct the metabolic hangover.